Hi everybody! Back at it again with a continuation of the Chapter 22 Immune System PowerPoint. And I am running out of steam because I have recorded like 5 videos today, so if my speech is a little bit not what it normally is, I apologize. My mouth is tired. I've been talking to nobody for hours and hours. Weird, I know, but part of my job. So, where we left off, we were talking about cytokines. So I'm not going to read this slide to you. It's just kind of a nice reminder slide for, hey, cytokines are a large and diverse group of chemicals. Here's what they do. And so I'm going to let you absorb this information on your own, since I went over it in a fair amount of detail in the last PowerPoint. And we're going to move on. So I mentioned in the last PowerPoint presentation that we were going to move on to humoral immunity, but since we spent the last presentation talking a lot about cellular immunity, let's just revisit a summary of how all of that works. Okay, so if we've got an infected cell, or a cell that is somehow abnormal, and keep in mind, typically these are regular somatic cells of some kind. But it's possible for cells that are members of the immune system to become infected as well. They're not bulletproof in that regard. So imagine a cell that has a virus or bacterium inside of it. That virus or bacterium is going to be bound to a class 1 MHC protein on the surface of the weird cell. CD8 T cells are going to recognize that, and they are going to initiate the production of cytotoxic T cells, which again are our warriors, memory T cells, the historians, and suppressor T cells, which are the peacemakers. So, warriors fight the war. Historians record the events of the war and stick around in case the antigen comes back. And then suppressor T cells are going to control or moderate the immune response. So they're going to keep the battle from getting out of hand, and they're also going to help ramp it down when the infection is no longer present. Activation by class 2 MHC proteins. Remember, these are bound to antigen-presenting cells and other lymphocytes. So class 1 MHCs are more ubiquitous. Ubiquitous is a word that means numerous or common, usually a combination of both. So when I say class 1 MHCs are more ubiquitous, that means they are all over the place. Almost every cell has them, whereas by contrast, class 2 MHCs are limited to a specific group of cells, namely antigen-presenting cells and certain kinds of T cells. So CD4 helper T cells are going to respond to foreign antigens in a class 2, and they are going to stimulate more helper T cells. So remember, these are kind of like generals in the army. So battle coordinators and directors. And memory helper T cells, which are going to work with the memory cytotoxic T cells to remember the pathogen for next time. Please also remember that helper T cells in particular are also going to be able to activate the humoral immunity side of things and initiate the production of, for example, antibodies in addition to the action of cytotoxic T cells. So let's move on to the humoral immunity side of things. So let's add something into this picture now. So helper T cells are going to be doing the activities that allow this communication and feedback to happen for the most part. So antibody-mediated immunity is different in that instead of directly attacking anything, the B cells are going to produce proteins which float around and stick to antigens. So let's start looking at that. So, B cells experience antigen sensitization. We'll talk about that. We're also going to talk about how they're activated by helper T cells, which is something I've already alluded to. Proliferation via clonal selection. And differentiation into plasma cells. So not all B cells are plasma cells, but plasma cells are the subset of B cells that actively secrete antibodies. So this is our jumping off point. Let's go look. So we'll talk about the other antibody classes, but the ones that are specifically on the surfaces of B cells, so I'm going to add surface, are IgDs. There are others that are either free and floating in the plasma or sitting on other kinds of cells, but IgDs are on B cells. So, what's the big deal with antibodies? They're just proteins. Why do they matter? Why are they important? Why is it bad if you don't have enough of them? Well, immunology is its own science and discipline, to be sure. It is, compared with other corners of physiology, not the most well understood, but that's because it's very, very tiny, and there's a lot of nuances and a lot of very specific little tiny details and steps and processes. This is, for example, why autoimmune disorders have not been solved yet, because there's a lot we don't know about the immune system, including antibodies. So there's millions and millions of different antibodies in different B cell membranes. So again, if we're talking about ones in B cell membranes, we're talking about immunoglobulin, that's what IG stands for, Ds. Not even worrying about IgMs or IgAs or anything else. So, if you do the math and look at the different parts, so we've got a heavy chain, which remember is just an amino acid chain. So there's 2 heavy chains and 2 light chains per antibody, making this Y shape. So there's 150 V genes, 5 J genes, and 10 different ways of joining the V and the J's together. So this is post-transcription modification, so alternative splicing. The total possible number of light chains is 7,500. The total possible number of heavy chains, probabilistically, is 2.4 million. So, if you draw this out by looking at how many possible combinations there are, you get 18 billion different possible antibody combinations. And remember, antibodies are specific to their antigens. So I'm not saying that 18 billion different antibodies definitely exist, but rather we have a possible library size of 18 billion antigens that we could conceivably remember. So this means our immune system's ability to learn about antigens and record them and remember them and make antibodies against them is very, very strong and very, very flexible. So that's what this number really means. It's that the possibilities of antibodies to create against conceivable antigens are extremely numerous. I said arrow. There we go. So, antibody specificity depends on the structure of the heavy and the light chains. And if you look carefully on the protein, you'll see that the binding site is at the tip of each part of the Y. So, the shape of the chain and the chemistry of the chain as well is going to determine the shape of the binding site and therefore which antigen the antibody binds. So there's 100 million different antibodies on either B cells or just in the body. So this 100 million is for both. And that's a lot. That's a really, really big number, which means that, in general, our immune system is pretty good at being specific. It's good at sorting and remembering and keeping track of a very large number of antigens. Okay, so B cell sensitization. Yes, there are antibodies on B cells, IgD specifically, but we're not fully abandoning MHCs either. So we're still worrying about major histocompatibility complexes. So those B cells have specific antibodies in their membrane. And that antibody, as per the specificity of the binding site, is going to bind to a specific antigen. So then the antigen is going to end up being taken into the cell and moved into a class 2 major histocompatibility complex. So remember, antigen-presenting cells and lymphocytes specifically express class 2 MHCs. So once we have antigen bound to antibody and antigen bound to MHC, now the cell is considered to be sensitized. So what does that mean? Well, sensitized effectively means the initial steps of activation, right? So we're starting to have humoral immunity be activated. So you can't have activation fully without sensitization. So a sensitized B cell encounters an activated helper T cell. So remember, I made helper T cells be the link between humoral and cellular immunity. So that's what's happening now. So those little reciprocal arrows that were linking those two legs of the diagram, this is what you're looking at when you look at those arrows. So once the B cell has its antigen displayed both in its class 2 MHC and on its antibody, then it's capable of interacting with a helper T cell. So the helper T cell is going to stimulate the B cell with cytokines, thus activating it. So basically it's going to hang on via the dock between the class 2 MHC and the T cell's receptor. And those cytokines are going to be released while the two are docked. So one component of this is close proximity. So we have this very, very small gap that the cytokines can diffuse across, and they can bind to the surface or inside of the B cell, and that wakes the B cell up. So before we move on from this slide, I do want to point out, even though it's not explicitly stated in this panel, that the specific T cell is the CD4 helper T cell, not the CD8. So refer back to that review slide from T cell activation if you want to look at the difference between those in summary. Okay, so after that CD4 helper T cell has activated the sensitized B cell, now we're cooking with gas. So T cell cytokines, so released by that helper T cell, cause proliferation. So proliferation you should take to mean mitosis, meaning an increase in B cell number. And those are going to differentiate either into memory cells, which, as you know, are inactive, so they don't participate in the battle. They're the historians. They're there to remember. But also plasma cells. And plasma cells are going to be the ones that are producing antibodies. So the antibody secretion rate is up to 100 million per hour. So the reason these are illustrated differently is because they look different. So remember when you were looking for lymphocytes in the blood slide, and some lymphocytes seem to have very little cytoplasm, others seem to be a little bit bigger and have more. Odds are the bigger ones were plasma cells. Plasma cells need to have more rough endoplasmic reticulum because that's where proteins are made, right? And so basically, plasma B cells need to have a larger antibody factory. So they tend to be bigger, and they tend to have a little bit more cytoplasm to make room for that additional endoplasmic reticulum. So that's the reason for the appearance difference that you saw. I didn't tell you initially because I didn't want to confuse you while you were just trying to learn your leukocytes, but now you know, and so now I feel like it's appropriate to tell you. Okay, antibody classes. I mentioned that IgDs were on B cells, and that is true, and I also said that we would talk about the rest of the kinds, so here we are. Most of your antibodies are simple Y-shaped chains. There are obviously some exceptions, right? And most of those Y-shaped chains are IgGs, so immunoglobulin Gs. These are super diverse because they're made contingent on recognition and sensitization. They also cross the placenta, and that imparts immune protection to the neonate for about 2 months until those antibodies are cleared from circulation. Hang on just a second. I'm going to go shut the window because there's some crows outside my window that are very mad and are making a lot of noise. Okay, I'm back. Turns out it's crow breeding season right now, and lots of pairs of crows have active nests hidden away. And then I looked out the window and there's a cat wandering around in the courtyard, making the crows nervous and angry, so they're screaming at it to go away. All right, back to antibodies. So, we'll talk more about neonatal antibody needs and protections as we move on. I just wanted to mention the placenta crossing ones. But also remember that anti-Rh factor antibodies can cross the placenta and begin to attack fetal red blood cells. So that's what caused hemolytic disease of the newborn that we discussed when we were talking about blood typing. And guess what? Among those, IgGs are those. So this is a double-edged sword. You want to impart protections on the neonate, but in some cases, those antibodies crossing the placenta can be detrimental to fetal health rather than positive. IgEs are on basophils and mast cells. We're not going to go into specifics of those, just know that they're on them. IgM, these are important for the initial phases of antigen exposure. So that first time that you're encountering something, for example. So we will look at a chart that shows you the relative expression of IgM and IgGs, for example, after the first exposure to something. So you'll see a time course plot of relative amounts of those two things. So just keep that in mind. But IgMs are a little bit more complex looking, as you can see. So they're kind of this pentagonal raft of antibodies all connected to each other. And so these are secreted following the first antigen exposures. Interestingly, these are also the variety of antibodies that anti-A and anti-B antibodies against red blood cell antigens are. Again, remember, I said nobody really knows how they get there. We just know they're present at birth, so their exact origin is unclear. IgAs are in secretions, so secretions specifically being exocrine secretions. So let's put a little asterisk there and say exocrine. So mucus, tears, saliva, semen, and breast milk, just to name a few. So again, I promised I would mention more about neonate protection. So the antibodies that cross the placenta impart protection and immunity to the neonate, so newborn, for about 2 months. And IgAs can also make it into the neonate via breast milk. So this is an additional step that can help impart protection to new people who have not had a chance to form their immune systems yet. So we looked a little bit of antibody structure previously, and I mentioned the heavy and light chain and all the possible combinations. So it's important to remind you that antibodies are not alive. They don't make choices, quote unquote, like cells do. They're non-living, so their ability to perform specific or very complicated immune functions is fairly limited, but given that they're not alive, they can do quite a lot. So Antibodies will bind to the antigen at whatever site on the antigen fits their binding site. And that's not going to be all of the antigen, only certain parts of it. In other cases, the antibody may bind to a complete molecule. Maybe, for example, it is a venom. So this is how antivenin works. If you get bitten by a rattlesnake, you're going to get injected with a serum called antivenin. What that really is serum that is rich in antibodies that stick to that venom and prevent the venom from doing its work. Also, antivenom is very, very expensive. The hospital bill cost for an average rattlesnake bite is like hundreds of thousands of dollars. So try to avoid getting bitten by a rattlesnake if at all possible. Interestingly, some antigens are only antigenic if they're attached to some kind of carrier. So these are called haptens. Haptens by themselves may not initiate an immune response, but haptens paired with a carrier, sometimes it's a carrier that exists previously in one's own body, that can initiate an immune response. So it really just depends. So urushiol, which is a plant compound present in poison oak and poison ivy, is a hapten. So when it complexes with certain things, it sensitizes you, and then you get a reaction to it, which is that horrible, awful, itchy rash. So I mentioned that compared with actual cells, antibodies have relatively limited capabilities, and that's true. So let's talk about what exactly it is they do. One of the things they do is neutralization. So in a lot of cases, the antigen binding site at the end of the little y corresponds to the active site of the antigen. So the spot on the antigen that makes it be able to be nasty in some way, antibodies can stick to and prevent the nastiness. So I mentioned antivenin, for example. So for a lot of venoms are typically complex glycoproteins, and those proteins have some spot on them that does something bad to our cells and tissues. So for example, hemotoxins are going to basically melt our red blood cells and turn our blood into jelly. So antivenin would be an antibody serum, and for that, the antibodies would bind specifically to the spot on the glycoprotein that causes the snake venom to be bad in some way. It can also cause agglutination and precipitation of antigen-bearing pathogens. So this is including foreign red blood cells, but also other things. It can activate something called the complement system, and you can actually see complement binding site here and here. So we'll talk a little bit more about the complement system later. They can attract phagocytes. So often stuff like this, which is an antigen of some kind studded with antibodies, is going to be very attractive to eosinophils, for example. Opsonization is increasing the effectiveness of phagocytes. So how does this work? Well, maybe the antigen is a bacterium, and the bacterium has a glycocalyx that makes it really slippery, so it's hard for macrophages or other white blood cells to hold on to. Maybe it's a neutrophil. In that case, the antibody might give the phagocyte something to grab onto. So over here we can see there's actually a binding site for macrophages on this particular antibody, which gives the macrophage something to grab onto, even if the antigen itself is slippery. They can also promote inflammation via activating basophils and mast cells. And in some cases, bacteria and viruses, their whole mode of infectivity requires that they be sticky and stick to our cells and tissues in some way. So in some cases, antibodies are effective because they prevent the bacterium from sticking, or they prevent the viral protein coat from sticking, rendering it neutral. So although antibodies are not alive, they do have a very large list of possible modes of action, and that makes them very powerful. If you combine that with their huge diversity of forms and very specific sites, that means that even though they're not alive, they're a class of molecules that matters a lot for human immune health and are really important. Okay, so here's that IgM, IgG activation curve response chart that I promised you earlier. So notice, here we have primary response and secondary response. I want to stress that this is to the same antigen. So, antibody concentration in serum is on the y-axis, so this is less, and this is lots. That's all you need to know. You don't need to know units. So, during the primary response, so your initial exposure, IgMs build up and begin to specialize to the particular antigen, and then IgGs follow a little while later. So this is time in weeks from exposure, so this is time 0. So you can see if this is, let's see, 1, 2, 3, 4, 5, 6, 7. So it takes a few days for your immune system to notice the pathogen, then it starts making IgMs. And then it takes about 2 more days after that for IgGs to get going. So there's a delay. This delay is why we get sick. So bacteria and viruses basically take advantage of that window in time when they can operate with impunity because the immune system has yet to notice and respond to them enough to defeat them. So notice the secondary response, this is day 0 from exposure. Now there's no delay. So IgMs get activated once again, but IgGs shoot up. So they're going to rapidly neutralize the antigen, and this is because of the presence of memory cells. So fast. Because of memory cells. And memory cells can persist for 20 plus years. So for example, I probably have some memory cells still bopping around in my bloodstream that, let's see, how old am I? 31? Okay, so yeah, like, you know, maybe I was like 8 when I was exposed to chickenpox or something. I probably have some crotchety old immune cell that still remembers chickenpox that's been around for my entire adult life. So the secondary response is a basic principle supporting the use of immunizations. So here's the way immunizations are supposed to work. You get a version of the pathogen that has been disabled in some way. So maybe the part of it that makes it nasty and makes you sick has been cut off by a lab scientist, or maybe it's been killed with heat, or something like that. So after you get the immunization, you do get a primary response. So you get this, and you get to make memory cells. But you don't get sick because the pathogen has been rendered incapable of making you sick. What that means is, after you get your immunization, once you are exposed to that pathogen again, now your IgGs shoot way up really fast, and you don't get sick because your immune cells remember that pathogen. So that's how vaccines work. So this is just a chart showing you sort of the time in weeks, and then the level of active immune cells. And as you can see, as far as the immune system is concerned, we have innate and non-specific defenses. So these are things like neutrophils and macrophages. We also have specific defenses. But everybody participates, just at different time frames and at different levels. So one of the reasons that we're not constantly becoming ill, even though the world around us is covered with bacteria and crawling with viruses, sorry if you're a germaphobe, that's just true. The reason that we don't get sick all the time is because look at this broad diversity of cells that are all responding to this same exposure event. And on very different timescales, right? So the immune system is really powerful for its diversity of response and for its specificity of response. So wrapping up the immune system, let's discuss forms of immunity. So immunity is just responding to threats to homeostasis. That's all. Innate immunity is just the general stuff that you're born with. So you don't have to have any prior exposure or antibody production. That's the key to understanding innate. So this is everything from neutrophils liking to eat bacteria to just having skin, which is a physical barrier to things getting inside of you. Adaptive immunity, this is the stuff we've been talking about this PowerPoint. So cell-mediated versus humoral immunity. This is a corner of immunity that is more specific, but it requires that your immune system learn about antigens. And the learning is the process where we get sick and then the next time we're exposed we don't, or we get a vaccine and the next time we're exposed we don't. Active develops in response to antigen exposure. And you can get naturally acquired active immunity or artificially. So this is just things like getting sick. Artificially induced active immunity is things like vaccines. I want to stress, we think of vaccines now as things that come in needles and that are very sophisticated and designed in labs. That's true. Didn't used to be, though. So the first vaccines involved, let's take, for example, the vaccine against smallpox. Cowpox is similar to smallpox, but unlike smallpox, cowpox does not kill you. It just gives you lesions on your hands. So it's transmitted zoonotically from cows to humans, so milkmaids would get it. And eventually, someone figured out, a fellow whose name escapes me at the moment, the similarity, and he experimentally took pus from cowpox sores, gross I know, and then put it on a needle and stabbed somebody else with the pussy needle. Yuck. However, that act conferred immunity to smallpox, and the person didn't get anything more serious than cowpox. That was the sort of origin story for immunization. So it's a lot less bad now than it used to be. It used to be that someone would have to stick you with a needle covered in somebody else's sore pus to give you immunity. So if you're gagging, I'm sorry, but that's true. Just look up cowpox. Okay, passive immunity. So this is transfer of antibodies from another source. So I mentioned, for example, the crossing over of IgGs and IgAs from either across the placenta to the neonate or through breast milk. So that's going to be naturally acquired passive immunity. There's also artificially induced passive immunity. So this is when there's some sort of a medical emergency and a population of antibodies from outside the person is introduced into their system to try and deal with the infection or the immune offense, such as it is. So, for example, the rabies vaccine is such a thing. So, you may have noticed that, among your childhood vaccinations, you have your measles, mumps, rubella, you have a bunch of other things like polio vaccine. One thing you don't have in your childhood vaccine suites are rabies vaccines. Why is that? If we have a vaccine against rabies, why wouldn't we want to give it to everybody? Well, the rabies vaccine is antibodies that are specifically designed to combat infection. So the rabies vaccine is not preventative. It is to treat an active or suspected active rabies infection. So it's an antibody developed in an animal that sticks to rabies. Another example of this is, as I mentioned previously, snake antivenin. It's also called antivenom, but it's called antivenin because the IN suffix acknowledges that antibodies are proteins. So what you're getting when you get injected or infused with antivenin is a bunch of protein. Those proteins are antibodies. They're going to go stick to that venom. So this is a summary slide summarizing all of that stuff. It is by no means informationally complete, so you should not memorize this and consider yourself to be done with the immune system. But, learning new things is hard, and especially when you have a really complex system like the immune system, finding a place to start can be challenging. So you can use this chart to figure out where you would like to start and then cross off parts of the chart as you feel you've mastered them. So that's what I recommend doing with this particular diagram. So one thing I do want to close on is the difference in infectivity and the response between bacteria and viruses. So bacteria, because they're large-ish, they need to first be eaten and then digested. So the antigen presentation component of bacteria is not the whole bacterium, it is just the piece of the bacterium that is antigenically useful. And then we get cytotoxic T cell activation and CD8 helper T cells, CD4 helper T cells, my bad. CD4. And then we get humoral immunity and cell-mediated immunity. So in both cases, antigen presentation activates both sides of specific immunity. Oh, I didn't realize I was drawing with the PowerPoint marker. Okay, so what about viruses? Remember, viruses are not alive, right? So typically, and unfortunately, what that means is they have to be either taken up by APCs, so I mentioned that very, very beginning of the first immune system PowerPoint. So sometimes APCs can just gobble up the virus directly. But more often, tissue cells are infected, and then they're going to display the antigen in their membrane. If the infected cell is an antigen-presenting cell, they're going to put it in their class 2 MHC. If it's the antigen presentation mode, we're going to activate helper T cells. If it's the appearance of the antigen in the plasma membrane of an infected cell, that's going to stimulate cytotoxic T cells and NK cells, which is going to destroy the infected cell. The infected cell is also going to release interferons, which allow neighboring cells to resist viral infection. B cells, meanwhile, are going to be producing plasma cells, and those plasma cells will produce antibodies, and that's going to result in the destruction of viruses that are floating outside of cells. So let me switch colors here, maybe to blue. This leg of things is deal with cells that are already infected. This is prevent, can't draw T's, future infection from viral particles that are cell-free, meaning outside of cells. Okay, and that is that. I would like you to read 22.7. I have a lot of recordings to make, so I'm not going to talk about them directly. But it's a relatively short piece of the chapter, so it will be represented a little bit on your test. We're going to focus way more on the stuff I talked about in detail. But this will address questions you may have about things like the effect of stress on the immune function, development of immunocompetence, allergies, and hypersensitivity. It's not very long again, but it is very informational, especially as it relates to things that students tend to have lots of questions about. So go ahead and read this, and then there's probably going to be some stuff that you don't fully understand. That's okay. Ask me about it in our Zoom session, and I'll be happy to answer. Okay, and that concludes the immune system. So I'm going to stop this now and begin recording the next presentation.